Good afternoon. My name is Olivia Koues, and I'm the Managing Director of the Advanced Genomics Core. Today, I'm going to discuss our newest spatial transcriptomics platform, the NanoString GeoMx Digital Spatial Profiler, commonly referred to as the DSP. Since our focus today is spatial technology, I thought a good place to start is by going over the history of spatial transcriptomics at the AGC. Due in large part to the popularity of our single-cell workflows, our 10x support team approached us during the summer of 2019 regarding onboarding their Visium platform and possibly becoming a certified service provider. We were indeed very interested and started working during the autumn of that year on coordinating what a spatial workflow would look like. As you can imagine, the platform would require additional skills in areas of expertise. We worked closely with tissue processing facilities around campus to get a handle on what it was going to take to expand into this field. I really want to stress how helpful Paul Harms and Ingrid Bergen, especially Pavlina and Ingrid's group, were in optimizing the tissue processing. We really wouldn't have been able to hit the ground as quickly as we did without their assistance. By the end of 2019, we had received our first Visium reagents and quickly completed our first project upon return from the holidays. In fact, we were able to complete 3 projects before the pandemic shut us down in March 2020. We were happy to see the customers quickly come back to the platform upon their return to research and worked most of the last year on expanding the business and streamlining the workflow. Highlighting our achievements, we were named a certified service provider for Visium in November 2020. Having established ourselves, we felt ready to tackle an additional spatial transcriptomic platform when approached by the single-cell spatial analysis program to house the NanoString DSP. The instrument arrived just before the winter break, and we quickly learned that installation and training during a pandemic can be difficult to coordinate. Training finally occurred in March 2021, but supply chain issues resulted in the first projects just recently being completed. Finally, we took delivery of the newest Visium reagents capable of accommodating FFPE samples just 2 weeks ago. So that workflow will be spinning up soon. As you can tell, the Visium platform is definitely more established on campus, and we've already heard about it today. So I'm going to focus on the DSP. While overall the workflow is similar to Visium, where you prepare slides, image tissue, make libraries, sequence the transcripts, process through a vendor pipeline, and analyze the data, the DSP's approach to spatial transcriptomics is quite unique. For the DSP, a series of imaging reagents tagged with fluorophore are used to visualize the tissue, and a series of profiling reagents are included for protein or transcript interrogation. These profiling reagents are either antibodies to detect protein expression or in situ hybridization probes to detect RNA. Initial processing involves hybridizing these reagents in a method very similar to protocols routinely used in IHC or ISH methods. 1 antigen retrieval, 1 staining step, and then the slide is ready to load on the instrument. So how does this work? Each slide is scanned on the DSP. Images are stitched together, so you have full-size 4-color images. Then the interactive software is used to pick regions of interest, a process I'll go over in a little more detail in a few slides. Once you have defined your regions of interest, then the instrument goes through and sequentially illuminates those areas in a very specific manner. Oligos from each illuminated area are collected and parsed into individual wells on a 96-well plate. These collections are turned into libraries for Illumina sequencing in order to quantify the abundance of oligo tags found in each region. The technology is based on the NanoString's unique probe architecture. DNA barcodes are attached to the protein or RNA probes through a photocleavable linker. This linker is the heart of the technology, as it allows you to use the instrument's micromirror system to optically dissect and subsequently collect the oligos bound to your regions of interest. There are many different ways to define your regions of interest. You can use geometric shapes. There's the ability to do phenotypic segmenting on the fluorescent image, which I'll go over in a little more detail on the next slide. You can also segment on non-contiguous tissue. For example, if you have cell-specific markers, you can mask based on a marker to isolate the oligos for an enriched population of cells. Using additional tools such as ImageJ, you can create contours around a structure of interest to obtain a gradient of expression. And then finally, you can take an unbiased approach and simply grid the entire tissue. Some of the most common questions for the platform are around size limitations of the ROIs and overall cell resolution of the assay. As defined by the objective lens and its field of focus, the biggest ROI allowed is 600 by 700 microns. As for the smallest, addressing the cell resolution question, in terms of illumination, would be a diameter of 10 microns. However, this is insufficient to obtain robust data. The minimum ROI size will depend on your tissue, the types of cells within the ROI, and their transcriptional activity. For RNA assays, you typically need an ROI size sufficient to capture 100 to 200 cells. But for protein assays, an ROI size to collect 20 to 30 cells is sufficient. A high-level overview of how the segmentation option works is shown here. Once you have your region of interest identified, shown in the circle, the DSP allows the user to create masks using the imaging markers and the light to dissect out the cell populations you care about. In this example, you would first illuminate the tumor using the criteria defined by the magenta label on the left and collect the oligos from the bound probes. The process is then repeated using a different set of criteria to release the oligos from the surrounding cells in the tumor and microenvironment, labeled immune cells here. When using masks, one region of interest will turn into multiple areas of illumination. Each area of illumination gets deposited into its own well, thereby creating two expression profiles for the same ROI. Once collected, the released oligos are subjected to standard Illumina library prep and sequencing. The NGS pipeline is relatively simple. The FASTQ files are trimmed, stitched, aligned, and deduplicated using a UMI. This is a pipeline run by the Advanced Genomics Core. Once the probe counts are generated, the data are actually pushed back onto the instrument for the user to analyze. The software walks you through all data QC, normalization, and initial expression analysis steps. As you have probably gleaned, the DSP platform is very flexible, which means there are lots of options and extensive planning that goes into designing an experiment. First, in terms of sample compatibility, you can use either fresh, frozen, or FFPE tissue. Due to how the ROIs are defined, you can put tissues of various sizes or from multiple sections or specimens on the same slide. Secondly, the DSP system is agnostic to the imaging reagents. We have lists of NanoString available antibodies, as well as those previously validated by their technology access program. But really, as long as the antibody conjugate works with one of the 4 channels on the instrument, it should be compatible with the technology. There are also options regarding the assays you can run. While the most popular NGS readout is the whole transcriptome atlas, covering about 18,000 genes, for human samples, there are other RNA assays, as well as the recently released protein NGS panels, where you can mix and match probes to design your own protein spatial assay. Finally, there's the selection of regions of interest, where you define the shape, the number per slide, and the strategy of how you want to interrogate the tissue. Fair warning, there is a significant customer time commitment the day of ROI selection. The DSP is very much a choose your own adventure, with the flexibility to design assays to fit your experimental goals. If you are interested in designing an experiment or learning more about the technology, please reach out to us at the Advanced Genomics core. With that, I want to take a moment to acknowledge the Advanced Genomics core staff for their dedication and hard work to bring on new technology during a pandemic. It definitely wasn't easy. Thank you.